have different approaches to share science. We have different national meetings, regional meetings here in Colombia, and now we are launch our RSG Colombia webinar series supported by RSG Turkey. And as speakers, we want to invite people working in Colombia universities in order to support our local network of bioinformaticians, and also speakers from different universities in the world to know different approaches in bioinformatics. And today we have the pleasure to have a really great talk by Gustavo Salazar. He's from Colombia, and now he is going to talk about a showcase of protein data visualization. So Gustavo, if you want, you can introduce yourself first, and please go ahead, and thank you for being here in our webinars.
Cool. Thanks, Yasid. Hello, everybody who is listening now. Yes, I'm a Colombian who have been studying overseas for a little while. I first did my master's in South Africa. That was in computer science. Then I did a PhD in bioinformatics, also at the University of Cape Town. And since 3 years ago, I moved to Cambridge, United Kingdom, and I've been working at EMBL EBI, specifically in the protein families group, and working with several kind of well-known resources in the bioinformatics community, such as PFAM, InterPro, the EBI metagenomics, and a couple of others that are around. What I want to do today is more like a path throughout different projects that I've been taking part for a while, and just kind of showing you different examples of visualizations that I collaborated with in bioinformatics. Some of them for my PhD, some of them as a side project, some of them here at EBI, but all of them have in common the idea of visualizing data and using the web as the platform, because, well, we know that nowadays the web is the main platform for any kind of communication. Hence, I want to just make a case on a common division that is happening between types of visualizations, not just in bioinformatics, but in general. Visualizations are being kind of divided in two categories: exploratory or explanatory. Exploratory when where the visualization tries to just offer different views for different types of data. They allow you to go through like usually big data, and you can go in different parts of it and just try to find what is actually interesting for you. And explanatory visualization can be a bit more classic view of visualization, when the kind of diagrams that we have in textbooks or that sort of stuff, trying to explain something on it. And those are also very important, and I will try to go through. Particularly in bioinformatics, we have seen visualizations in the exploratory side from the moment we get data, from the moment we get some sequence that out of the sequencer. We are starting analyzing that. We know that a sequencer is going to give us so much data that we cannot just read it manually, and we need to have some sort of view on it. For instance, we explore all the reads that give us the alignment that we explore there. Once we have that, we have other tools such as Ensemble, where you could now not only have that data, but now seeing it in a higher level, meaning now it's not just the reads. Now there are features in that whole chromosome, and there are features that are like, for instance, the gene, or more specific, like pieces of that gene. Obviously, another kind of exploration is a statistical exploration, where you have the big data and you try to get aggregates out of this data, make clusters, or make what we always try to do, make sense of this data. We are exploring this data in different ways. Now that you have that data, when maybe we jump in bioinformatics, and from genes, then we start talking about proteins, and then we can have the typical track visualization in a protein and try to say features similar to the kind of domains that we can see on PFAM. And even in a higher level, then you have things like interactions between proteins, and this is another type of data, and how they communicate, they not communicate, they interact, and how that is important to try to discover pieces of information out of it. On the other side, on Exploratory, we can have these amazing graphics that some very talented artists are giving us. Maybe some of you have seen this sort of visualization. There's a famous video by Drew Berry, who just try to simulate interactions that are happening in a intracellular level. When you see these videos, they are amazing. They kind of like this little molecule here actually seems that is walking through the path, and that it gives a very interesting perspective. It's not so realistic. We know that things are not that clean. We know that in between the different components is many noise, but it serves the purpose of explaining a procedure, and that way it helps us to go through, to understand something. We are explaining some of that. Another example in bioinformatics that I saw recently was showing the evolution of the Zika virus around the world, how it moved from region to region, and the different mutations that was having in different countries, and how until the final epidemic in Brazil. So it's all of that. It's another type of explaining a case of how from the starting point to the end of that evolution of that particular disease. And then back to the ones that I was explaining on textbooks, and how they, we learn based on this. When we are going into Dogma Central or anything biologically referent, we take these books and we learn based in these visualizations. They might not be exact, they might not be precise, but they serve the purpose to help us understand. Recently, there have been a push on visualizations that are kind of in the middle ground, that you have a bit of exploration, but at the same time that you are exploring, you are actually understanding some of it. So that's another type of visualization there. Now that I got through this introduction, unfortunately, my whole career has been in this side, in the exploratory side, and I'm going to start talking of a couple of projects that I have taken part and show you the project. My background is, as I mentioned, was more on computer science, and therefore my emphasis will be on the tool. So sorry for the pure biologists, but I will try to keep in mind the biological cases on all of this. But yeah, I'm going to focus a bit more on the creation of the tools. Sorry. Okay, sorry, just double checking I was still connected. Okay, so the first tool I'm going to talk is actually one of my first introductions to bioinformatics. When I arrived to South Africa, a good friend of mine, Rafael Jimenez, was working in this tool called Dusty. He showed me the tool that he was working on, and he needed to give an extension. Yeah, I collaborated with him on that particular case. Dusty basically takes one protein ID, so a UniProt ID that you are interested in. You put it here, click on go, and Dusty collects information out of multiple servers. Put them all together. Mostly this information is about the protein in a positional mean. So like knowing there is, for instance, a beta strand in this position, and it's another beta strand in this position, and so on. There are many features that are tagged in there. The idea is that multiple projects can have their data, and that it can take that information from the different places and put it in a single place. When you see the information here, this one can be filtered by things in that side. For instance, filtering by the ontology, so the kind of feature that they are there. So all those features have been annotated in an ontology. And for example, the beta strand is one of the regions. And then if I don't want to see them, then I click there. The component that I was collaborating with was only the 3D structure, and was something very simple. I was taking an existing component that was created in JMOL, put it in this website and making it interact. There was another server that I could map coordinates from one side to the other, and if I click in one, I will highlight in the structure. The idea behind Dusty was just a simple client using what's called the DAS protocol. DAS stands for Distributed Annotation System, and the DAS protocol just allows this idea of getting data out of different places in an organized way. So I'm trying to show this timeline here on different servers and how the client, Dusty, will put that all together. So if in a given point, somebody opened Dusty, the first thing that Dusty does is go to the registry and, hey, do you know which servers are there for proteins? And then the registry returns a list of those sources for proteins. And now there are sources for proteins. Okay, the user can put its query. Okay, I actually want information for the A4 human, and it goes to that reference, and then that server returns the sequence. And now that you have that reference, then you can ask all the other servers, hey, who else have information about A4 human? And one or many resources will return all of that data in here. The advantage that DAS offers, it was a very strict language. So when you were doing these communications, all of those were normal internet communications, HTTP communications, the same way that you write www.google.com. That's what this client asks to this server. But then the responses were predefined in XML language format that was predefined. So the client only knew how to process one type of format, but because all the servers were replying the same format, then he could process all of them at the same time. That project in 2008 basically was developed in JavaScript. It uses a library called Prototype to make the IJAS code and some DOM manipulation. It got very criticized, that library, because prototyping in JavaScript is dangerous. When you overwrite certain things, certain methods or certain behavior of the main types, then it is problematic. You cannot ensure any more things to work together from then onwards. But yeah, that's what we were using back then. And basically the trick or the visualization consisted in taking that information that came in the XML and transform that into div elements on the web page. Just was well styled, and then the div was in a specific location, and then we make sure that everything was aligned. But at the end of the day, we're just div. For the 3D structure, we were using JMol, which was a Java applet. For those of you that are all know, you know all the issues that we used to have in any web page that we're trying to use Java applets. And there were many of them. Then, because of security reasons between servers, in those days we couldn't just directly talk client to server. We needed to have a proxy. We needed to have an intermediary to make these connections. And in those days, that piece of code that we have defined was CGI written in Perl. That code is still available in SourceForge project Dusty. But obviously, I mean, if I'm describing all of this was quite old technologies. We did an effort in 2011 to rewrite the whole tool. Jose Villaveses was in charge of that project in that moment, but a couple of us, given that these projects are open source, we collaborated and we helped in some parts of it. And yeah, 2011 we was now JavaScript, but at least now was almost a new version of JavaScript, not completely, but mostly better written JavaScript. And we were using now jQuery, which was an improvement prototype. And for those of you that are web developers, you know that still jQuery have many caveats and things. And from those days to now, we have evolved JavaScript dramatically. But the tool that we were using there back in those, it was really good. And jQuery was very important in web development in general, to actually give power to tools that were happening or components that were running it directly in the client, among other things, because unify the way that we were doing the calls to other servers. So now when we were writing Dusty 3, our calls were throughout jQuery. And then that means that these kind of calls will be running not only in your browser, but in different browsers like Internet Explorer, Chrome, Firefox, all of them. So it will be a matter of compatibility. But basically, the drawing or the visualization itself was still sequence features getting transformed into divs. SVGs were starting happening around that day, but then in that moment we decided to go for divs just because it was more compatible in the browsers. The browser, the 3D viewer was still a Java applet, and now our proxy was an optional thing because some of the browsers now, some of the servers, sorry, were providing the standard that is now called CORS, and that standard allows that communication. Not all servers have it, and sometimes you have to do this trick of creating a proxy that would be the one interacting with it. But now, in that moment, in 2011, and from then, it was now available that client can directly connect to different servers. The repository of that code is also available here, if anybody was interested. I will share the link of these slides now in the tool. So I'm talking about Dusty, and what happened with it in history was a nice growth. This mark here shows the different versions that were exposed. And you can see it was growing nicely, and people were using it. And there was an effort for standardize certain things when 1.6 arrived for that version, and that's probably the zenith that we reach with Dusty or with DAS in general. When I'm talking about around 1,200 sources, those are where the providers of information. Not all of them were proteins, some of them were for structures, some of them were for genes. So the idea with DAS is it was just this standard, and there were different types of data that we will be able to deal with it. But still, there was many sources, and all those sources were things like UniProt or things like the NCBI. Some of the projects in the NCBI, they were sharing their data using DAS. In parallel, there was another project trying to push DAS 2.0, which tried to use newer standards in it, but it failed because the standard or the proposed format that they were using in DAS 2.0 was too different to the ones that were here. So for those hundreds of servers that were already implementing something, it meant a lot of work to be able to show it now in DAS 2.0. And then you might have noticed that this graphic is only written until 2011. And that's why, because actually from that point on, things were starting going down. I think the technology behind moved faster than the community that was behind us was able to keep up. And these kind of projects, if you don't push for changes, then they stall. And actually, by August 2014, ABI made a big announcement of retiring most of the supported things because of basically money reasons. It was the whole point of they were supporting servers that people were not using anymore. So, and this was the peak, and then it started going down. EBI supported this for a few more years, but given that less and less sources were providing information, then EBI also stopped giving support for this. And not only EBI, but the registry that it was running in Sanger back in those days also disappeared. And then many of these things disappeared. And unfortunately, well, there were not more servers about it. And that's why I cannot just show you a link of Dusty running, because, well, if there are no servers providing information, I cannot have a client showing that. So unfortunately, that's the case of it. Just want to check here. Yeah, just checking that everybody is around, that is still okay. That was the first part that I want to talk about it. There are, I think, 4 or 5 more tools that I want to talk around. But if any moment you have a question, just please write it in the chat. I'll have a look every now and then and try to solve any of your questions. Okay, then I'm gonna go back to the presentation now. Okay, and another project that I was involved between 2011 and 2013 it's called Byojs. Some of you might have heard about Byojs. This is basically an idea to create a library for components. So a lot of us developers are creating web visualizations, and then we started noticing that the effort that I was doing in one kind of visualization, it was getting replicated by somebody else. So people keep rewriting their things over and over. In that case, we decided to create Byojs back in 2011, and with the main goals of reuse data, maintain data, and deploy data, that all of these tools will be easy for the creator of a component. So if somebody creates a component to display expression on what part of the human body gets expressed, and then somebody else could just reuse and doesn't have to redefine all of this. You can see this kind of snapshot is a bit outdated, but it's exactly my point. This is what we created 7 years ago. Again, Byojs was an open source project, and it started here at EBI as well. The technologies that were involved for the registry, or for that web page that people can go and check what to look, if you are a developer, you'll see it is weird to mention Java, but that's what it was in that moment. You have to compile Java to be able to create the website. The components themselves, well, those were mostly JavaScript. You have to, when you were writing a component for Byojs, you have to put documentation using JStock. And yeah, style and HTML was based on this. But the big thing on this version of Byojs was that there was a very strict set of rules that you have to follow. Methods have to be written in one way, attributes have to be written in a specific way, same for events. And when you're gonna declare dependencies, there was a very specific way of doing this. But this was problematic because if you wanted to include a component into the registry, somebody have to recompile this with the new component. And there was a very manual task. Somebody have to go to things, update those files in the server and put it up there. So this was very slow to actually publish. And in 2014, there was a big push to create a new version of Byojs. In this new version, we reuse like the registry that was already available for JavaScript packages that is called npm. So there is a registry there that if you create a package, it could be a visualization or not, but you create something in JavaScript and you wanna publish it for people, then you just put it on npm, you publish this data in npm. So Byojs moved into that. And in 2014, the new version was, instead of people sending us their components, so we compile them into the project, now a new developer only have to just publish on npm, and we just check the npm registry and check if it has a tag that says Byojs, and if it does, then we display it in this list. The advantage of it is that now it would be a lot easier for people to publish into Byojs, and yeah, it makes that side a lot simpler. Downloading is also easier because now the data is already in npm, and then you can just directly download it out of there. The only thing that we kind of lose in that was a bit of the control that the components were having, or the developers have to follow these kind of rules on creating documentations, on creating the way to display a demo. So now it was a trade-off. We made life easier for developers, but now it's a bit harder to actually have a consistent documentation or have a consistent set of demos for each of the components. And since then, things have evolved a bit. This year we did during the Google Summer of Code, a couple of developers took on the task of renew the website, but the logic behind still being this idea of taking data out of npm. And if we, for the demos of it, we use a tool called Proserify, which packages these pieces of, if a component have dependencies, Proserify will bundle them together. And then using GitHub pages and Jekyll and other things, then we can automatically create the Byojs repository. The components now are free to develop in JavaScript. You can use whatever techniques you want in JavaScript, whatever sublanguage that you use. You want to use TypeScript, you want to use React, you want to use web components, all of that is valid. as long as at the end you publish that on npm with the tag Byojs. So it means it's a lot of freedom for development. Okay, I'll go again to the webinar. Just checking if there are any questions. Okay. Then, okay. If not, I'll keep going with the talk. Sorry. Okay. The next tool that I want to talk about was one that I worked on during my PhD. It was basically a protein-protein interaction visualizer. The idea is that a lot of the visualizers that were out there, like Cytoscape, there were tools that you have to download, you have to install in your computer to try to work on. And the ones that were actually on web were like using Java applets or things that were not natively web. So what I wanted to create is this tool that being able to play with this data on the web and then give the users the option they can upload their data and put it up there. This was published in 2014, and well, you have a section in that corner where you can put your proteins of interest, and once you click on one protein, then you have the graphic in this side that it will display those proteins. You can create rules saying in this case, I want to color by functional class and all the proteins. So that's why we have these colors here. And another one is show the label gene name, so a specific parameter of that one, only for proteins with a number of interactions higher than 2. So that's why these two are the only ones showing the label. The idea being is you can create your own rules, and that way you can customize the view or your interaction. If you want another view, then you click in this side, and then you see the circle view. And in the circle view, well, it's similar to the circles that has been widely used around, which for some cases might give other insights on the data. Some cases is as complex as the not link view, but at least it's an alternative. And one more that I developed in there was kind of a heat map that you can see which interacts with which one. So you can just do the links in there. I think because of time, I'm not gonna go and play with the tools themselves, but whoever wants to, they can use this link in there. I forgot to share the link of the talk itself, so I'm gonna copy this link and put it up here, so if somebody wants to check on it. Okay. Another couple of things that over there, as I said, the user can upload their own data. There is a format that we define on how the PPI data should be introduced on it, and how there's two files, one for the interactions and one for the features that you want to attach to each of the proteins to show. And we have already put a couple of species that we were interested in the lab that I was doing my PhD. So for the guys in the lab to explore these data sets, but other people have uploaded their data and have put it there. So it's things to work in there, things to play with. Okay, the technologies I use for this tool were mostly is JavaScript. It uses jQuery as the library to do things, but for the graphics it uses the D3 library. So if you are starting to thinking visualizations, I highly recommend the document-driven data library. It's really amazing for a lot of visualizations. The data itself was stored or is stored in Solr, and we had a small server that was more for kind of security, like so just allowing certain kind of queries into the Solr server, to the Solr engine. But also it helped us to have a concept of private and public networks. So in PIN, you could put your data in there and we will upload it in Solr, but it will be only the people with the link will be able to see that, and it won't be published. And so just to give that option of private data sets. Yeah, the code for the client is in this URL, and the code for the server is in here. Most recently, here at EBI, we have started working in this project called Nightingale. Before I go there, I'm gonna check again if there is any questions in the chat. All right, not so far. Cool. The Nightingale project is that same idea of having a track-based visualization, and basically the only new thing on it is the technologies behind. A lot of these visualizations are using many dependencies that are not standards. So some projects are currently using, for instance, Angular, and when they try to share it to a different project, then Angular gets limited, and then I will have to embed the whole Angular to be able to show. And that was one of the main criticisms with jQuery. Every time I was using jQuery in one of these projects, I had to upload the whole library, even if I was just using 10% of all the methods that were included in there. So it's one of the things that were a problem in those days. Things have evolved. JavaScript has gone from ES5 to ES6 and 2015 plus. And with that, the concept of modules and making JavaScript an option to actually develop big range applications then has actually grown a lot. But with that, a lot of those dependencies have appeared. NPM is the fastest growing repository of components of like in comparison with any programming language, and that means that shows the interest of people on JavaScript, but also shows you start getting attached and more with dependencies. So the effort that we were trying to do with the project Nightingale is to use web standards that are fully supported for browsers, or at least will be supported in the near future. And the one standard on JavaScript that we can see that going in that direction is web components. So web components allow us to pretty much write your own kind of HTML tag. So instead of the tag div or instead of the tag image, then you create a new tag. And what you see here are different tags that we created. And this is the tag for a navigation component. This is the tag for the sequence bar. And each of those are tags for tracks that we can play with. So now when somebody can use this, it still needs to include the JavaScript for the new tags. But when you write the piece of code for it, they look like just HTML tags, which is kind of the effort. And that's on the technical side. But on the more kind of social side of it, this project was born because we noticed that the guys from InterPro were creating a feature viewer for proteins, while we at InterPro were doing the same, and some guys on PDB were doing the same. So we just said, let's try to make a library of these things that are common, at least for the three of us, and work with it, but try to make it as standard as possible and as basic as possible. The way that if somebody else needs this, they can just take this component. So you can see the list of components here in this URL. The idea of starting with a track visualization was taking an existing project that InterPro uses called ProdVista and try to make the same functionalities that ProdVista have, but now on web components, and then making it a standard. Yeah, we initially was InterPro and InterPro, and then now some guys from PDV are joining us on this. And yeah, the idea is now it is a freely available set of components. And if somebody else needs a track, then can now inherit from our definition of track and create their own starting from there. All the code is available in there. On some of these components, we're still using as a dependency D3, but again, now D3 and newest version have been split into modules, so we are only using exactly the pieces that we need for it. So we need scale, we need zoom. So those things we take, and we don't have to upload the whole D3, we just upload those little things in there. And if any of you is a web developer, you know that nowadays we have to work on web environments, and the list of dependencies on the development side is quite bigger than what the user ends up actually having to see. But I mean, I'm not gonna go into those details now. Okay, one last check on this side. Checking out time, I'm actually running out of time, so I'm gonna go a bit even faster this last part. Now most of my time is dedicated to work on InterPro. Quick idea of the data that we have in InterPro. So when there are multiple proteins, we try to find these regions that are common on all of them. And in the group, we have curators that not only say this is common, but they actually annotate that region, and they say that this is a domain that is related with a biological process or is found in a specific part. So there's a lot of annotations included for a domain. And then there's these ideas of like creating a domain library. So all these regions that are common are saved in a common place. And the reason for this is that the moment that you have a new protein and you don't know anything about protein, you just have the sequence, then you can run tools on it and try to match any of these domains into it. And then if the match is perfect, then you can infer that the properties that were written for this domain here are actually valid in this side and the same for this domain. So now you know a lot of your protein based in this domain matching. We at InterPro don't define particular domains or families, depending on it. What we do is we take all these annotations that different people have done. There are many projects that are working on that using different technologies. Some are using hidden Markov models. Some are using some kind of profiling system, even patterns like kind of regular expressions, that kind of thing. And what we do in InterPro is to take all of those and create an aggregation of it. So those domains that Pifan said that exist for that protein, and they are, for instance, there is a domain in Pifan, and we have a corresponding domain in Panther and a corresponding domain in CDD. So the three of them align for the same and telling us the same thing, even when they use different techniques to get to that point. Then all of those we group into a single InterPro unit, and then we provide the users with InterPro scan software that allows to do this last part here, where you can have your new sequence, and we run not a single piece of software, but we've run all the matchers for all of these databases. Now we have all this information in our database, and we want to show it. We have a website for it, and what I've been working for the last couple of years, working on it for the last couple of years, is creating a new version of the website. The idea now is that you could access these kind of annotations from different points of view. In this slide, we are just showing entries, protein structure, and organism, and then you can just say, okay, no, I want now proteins that have a match in PFAM that are present in a specific organism, in, I don't know, gorilla or something like that. And then you can start narrowing your data, and all this, the result page here can be always being filtered further and further until you find exactly what you get there. We'll ask you to actually play with it. If you, for any reasons, are InterPro user, if you have to analyze data, go to the beta side of it and let us know if you have any issues, because that's why it's in beta. We have worked on it in a couple for the last 2 years, but we want to, keep improving and get the website in a stable position. The website includes a lot of other types of visualization. For instance, every time that you see the protein, you're going to see all the matches of the different interactions using Nightingale. Or when you see families that are in common, we use a similar visualization to the network, protein network that I showed early. And when we see a taxonomy, we can see a taxonomy tree that we can expand and work on that. So all of that data has been stored in an API, so the users now can just create their own scripts and get the data out of our API, and programmatically get all of that. And yeah, that's what I want to say on InterPro. This is the technologies I have in there. We put our data in MySQL, but all the links between the different pieces of data is in Elasticsearch. The API is written in Python and Django, and the client is a React application using just the latest JavaScript version, and yeah, many other components there. Just because of time, I think I'm gonna leave in there. I was only one more tool that I had in the slides, but you can also just check there. All the things in the slide have the links for both the code and the places that you can play with it. And for now, I think I'm gonna just leave it there and give a few minutes for any questions, if there is any.
Hi, can you hear me?
Yeah.
Okay, it's time for questions. Is there any question there? Okay, I have a question. So thank you for the talk, great talk. I think that is really nice to know for us as a biologist, to know this is the behind the scenes of the tools of we use daily. And I think that is a big effort from you, from your team, in order to retry the biological meaning of the data, And I would like to know what's your opinion in terms of what are the main challenges that you find in biological data in order to develop different visualization tools. I mean, in terms of the heterogeneity that you can find in the data. What are the main challenges that you find?
The thing is exactly that, and I think in biology we have we try to make sense of all this data, and we try to find the common places around it that we can work for it. But you always have to deal with the exceptions. is biology. Everything is based on exceptions. We have life because of mutations. So there is not just rules. And everything is something. So when we are trying to create these visualizations, I think the effort should be on finding those exceptions. So we try to visualize the things that are a bit different and highlight that somehow. You take two genomes and you compare them of the same species, and it's 99.9% the same. It's just those little changes that actually make sense. But try to put all of that in a single view. It's really hard, and I think that's kind of the main challenges, and that's why visualizations have to be interactive. A single image is seldom enough. An image needs to, a user needs to play with it, needs to like filter and being able to get to a point where is the actual data that they are interested. And the hard part is make that in a way that is powerful, but also easy. So people needs to.
Yeah, that will be a really cool combination between powerful and that. Okay, Gustavo, thank you very much. Is the last opportunity to make a question to Gustavo. Is there any question? No questions. Okay, so cool. Thank you again. And if anyone is interested to keep in contact with Gustavo, I think that is possible to send an email or
just put it there.
As some doubts to solve some doubts. Okay, thank you, Gustavo, and cool. See you next month in our next webinar. Thank you very much. And
cool. Thanks, Jesse. Thanks everybody for listening to me. Bye bye.
See you.